Hello and welcome to Science Matters from the Pacific Northwest Research Institute. Science Matters features discussions with scientists from around the globe on exciting research topics in genetics, genomics, and evolutionary biology with important societal impact. Today, we have a special seminar in recognition of Newborn Screening Awareness Month, which will feature speakers who will focus on the history, development, and challenges of newborn screening in the United States and Latin America. We have an exciting lineup, so I want to get right to it, and I will keep my introductions very short. Our first speaker is Dr. Jane Buckner. Dr. Buckner is currently the president and a faculty member of the Benaroya Research Institute here in Seattle. She's also an affiliate professor in the Department of Immunology at the University of Washington, and her laboratory focuses on research identifying the underlying mechanisms by which the adaptive immune response to self-antigens become pathogenic in the setting of human autoimmune disease. However, we've asked her here today to join us as a special guest because she is also, in addition to her own accomplishments, the niece of Dr. Robert Guthrie, who developed a blood test for PKU that was the basis for the first newborn screen. Jane has kindly agreed to give us some personal insights about her uncle, who played such a pivotal role in establishing what is arguably one of the most successful public health initiatives in the United States. Jane, if you'd like to share your screen and start, welcome.
Absolutely. Yeah, thanks for having me. Assuming my screen is being shared right now. And, it's a pleasure to join you. And I can't wait to hear about the status of newborn screening. This is a little bit of information about Bob Guthrie. He was born in Missouri, but actually grew up and lived in Minnesota, which is where I'm from. And then went to medical school and then ended up living most of his professional career in Buffalo, New York. But did end up moving to Seattle near the end of his life, and about the same time I moved to Seattle. And he's known for inventing the bacterial inhibition assays used for newborn screening. But of course, why am I talking about him? And that is because he was my uncle and played a pivotal role in a lot of ways. And well, I'll talk about that in terms of even what I do. So he's my uncle. I have a picture of his wedding to my Aunt Margaret. My mother is the one with the dark hair, and my Aunt Mary Lou is the redhead. And our family played a role in his development of newborn screening because Mary Lou's first child, Margaret, was born in the late 50s and was diagnosed with PKU at 15 months of age. Uncle Bob had actually done some work with PKU in the lab before Margaret's birth. And once he realized how hard it was to do the screening, that's what really encouraged him to apply the tools he was using in the lab to develop this first newborn screen for PKU. So that was all going on at the same time as the picture that's next to the wedding picture, which is my family, and I'm the baby. So in the early 60s, Bob had developed this newborn screening. And one of the things to know about him is he was absolutely passionate about the newborn screening. And as I grew up, he and his family of 6 children with my Aunt Margaret would travel not only around the US, but around the country, around the world, for him to share the news about newborn screening and how important it was. And so I have a picture in the lower panel on the left of Bob and most of the family visiting my house. That's the lovely garage of my childhood home. But what I thought was always the memory I had about them was that they would come to Minnesota to visit, and they would roll out of their VW van, all of them, and of course, fill our home with a lot of activity. And this was just a picture that we were able to find with the van in the background and a typical meal for the Guthrie family. So I think imagining the energy and enthusiasm it took to pile the whole family in the car and travel around. And of course, his family members were always huge supporters of what he did. And thinking about this story, and here I am, a rheumatologist, immunologist, and I primarily study type 1 diabetes. But what has been very interesting as we think about, actually about 80 years from the time he married my Aunt Margaret, but all the years with newborn screening, is that today in type 1 diabetes, we actually can predict development of diabetes. And one of the tools we need to use for that is to understand if individuals are genetically at high risk. So the idea of newborn screening started so many years ago, may well apply to the field that I'm personally passionate and committed to, which is identifying ways for us to identify children early in life who are at risk for type 1, because we now have interventions that could potentially delay or predict disease. So years ago, Uncle Bob was the guy who always had a camera in front of him, was referred to as the Cyclops because he photographed so many things, was passionate about boats. And then it turned out our paths crossed when I entered medicine, and without realizing it over the years, well, his work is influencing the work that I'm doing. So a real pleasure to always to hear other people talking about where newborn screening has gone to and how important it is. And certainly for my family, where we've had an individual profoundly affected by PKU, my cousin who I grew up with, it's really clear how important newborn screening is in changing lives. And so I appreciate that work personally as well.
Thank you, Jane. That was wonderful. That was really lovely. Just to remind people, we will take questions for all the speakers at the end of the session. Now, without further ado, our next speaker is Dr. Nina Gold. Dr. Gold is a board-certified pediatrician, medical geneticist, and medical biochemical geneticist. She's also the director of prenatal medical genetics and associate director for research at Mass General Brigham Personalized Medicine, and an assistant professor in the Department of Pediatrics at Harvard Medical School. Dr. Gold received her medical degree from Harvard Medical School and then completed a combined residency in pediatrics and genetics at Boston's Children's Hospital. She also trained as a fellow in medical biochemical genetics at the Children's Hospital of Philadelphia. She's the recipient of several awards, including Outstanding Resident Teaching Award from Harvard Medical School and the Shire Genzyme Award from the American College of Medical Genetics. Her current research interests include newborn screening and the identification of actionable genetic disorders. And we're excited to hear about some of her work. Nina, thank you for joining us.
Great. Thank you so much for having me. Let me go ahead and share my screen. Okay, great. Can everybody see that okay? Okay. Well, what an amazing story, Dr. Buckner, and that leads really nicely into what I'm going to be speaking about today. As you heard, my focus really now is on newborn screening, and in particular, using genomic sequencing to expand the number of treatable disorders that are screened. So I'll be talking today about the road to genomic newborn screening. We'll be first covering the current state of newborn screening in the United States, then discussing views on expanding newborn screening using genomic sequencing, and then finally talking about how data from biobanks and electronic medical records can be used to inform genomic screening characteristics like its specificity and sensitivity. So first, you've heard a little bit about the early history of newborn screening from Dr. Buckner already, but of course, newborn screening initially, came to be with Dr. Guthrie and the introduction of population-wide screening for PKU. And over the decades, many additional disorders have also been included. Several inborn errors of metabolism like galactosemia, maple syrup urine disease, hematologic disorders like sickle cell, and then multi-system disorders like cystic fibrosis, and endocrine disorders like congenital adrenal hyperplasia all made their way onto the newborn screen before 2000. And since that time, we've seen the addition of other disorders, as well as the addition of new methodologies for newborn screening, like population-wide hearing screening, and the addition of inborn errors of immunity, like SCID, and screening for things like congenital heart disease as well. Most recently, we have had some additions such as Pompe disease, which are extra challenging for us in clinic because sometimes the combination of biochemical and genomic screening that's used actually identifies children at risk, not for the infantile form of disease, but for the later onset or even adult form of those disorders. And so the paradigm of newborn screening is beginning to change a little bit. Historically, the criteria that has been used to guide the addition of disorders to what we call the Recommended Uniform Screening Panel in the United States, which is a federally recommended panel of the disorders that each state includes on newborn screening, those disorders have been guided by the Wilson-Jungner criteria, which was also developed in the 1960s. So many of us are already very familiar with these ideas, but basically, here what we're saying is this should be an important problem that has a clear treatment. We should all agree upon, who to treat and make sure that the treatment is available. And we should really understand these disorders quite well. And then there's sort of, additions like the total cost of a finding should be economically balanced as well, which I think we've begun to stray away from as we've begun to select some of these more rare disorders. So at present, the Recommended Uniform Screening Panel in the United States includes 37 core conditions and 26 what we call secondary conditions. And those secondary conditions are disorders which typically share the same biomarkers as the core conditions. They're not the target of screening, but can be kind of incidentally ascertained by the same screening methods as we're using for the core conditions. The interesting thing about this is that there are now a hugely growing number of additional genetic disorders for which there are targeted treatments. So, there are databases such as the RX Genes database developed by David Bick and others that have done an incredible job enumerating the wide range of disorders for which we have some sort of treatment. This doesn't even necessarily include expert management guidelines and surveillance, but true therapeutics that can be offered, either an early stem cell transplant or some sort of medication or dietary intervention that can fundamentally change the trajectory of this disease and improve quality of life for affected people. So right now, in individuals who have disorders that are not included on newborn screening, it can be a long road to diagnosis. So what sometimes happens when a disorder isn't included is that a baby can be born and appear perfectly healthy in the nursery. Then over time, they may miss milestones or develop symptoms of the disease, and eventually they might be referred to a medical geneticist or to another specialist, as a number of our colleagues are now sending genetic testing for a variety of disorders. And then as they wait for that evaluation and they wait for the results of testing, months later, they may finally be given the appropriate diagnosis. And so the problem with this paradigm is that sometimes when people don't know what they have or what they're at risk for, people can face dangers unexpectedly. So these are sort of a few stories that are clipped from the headlines, but you can see here, for example, a teenager in London who had a protein shake and had what's called OTC deficiency. So that large protein load predisposed him to hyperammonemia, which can cause neurologic sequelae and can be extraordinarily dangerous. Or a girl who was at risk for a genetic cardiac arrhythmia and didn't know it, and unfortunately suffered sudden death as a result. But the aim of expanding newborn screening to include more genomic disorders is this idea that when we know more, we can do better for these children. We can improve their pediatric health care. And it's not necessarily just following along the sort of Wilson-Youngner paradigm of applying an emergency intervention right in the neonatal period, but sometimes just improving pediatric health care or even lifelong health. So when we know more, we can apply gene therapy early on in the course of a disease, maybe before somebody manifests any initial infections and gets sick. Or we might be able to at least monitor them for the early signs of disease so that we can decide when they might need something like enzyme replacement therapy applied. So what do people other than me who study this and think about this all the time actually feel about expanding newborn screening with genomic sequencing? This matters a lot, especially when we're talking about implementing this type of technology on a population scale. There are a lot of different stakeholders and many people and groups to whom this would matter. Each disorder that's been added to the recommended uniform screening panel over time has had, a number of groups supporting it, people opposing it, and each disorder has been carefully considered. So if we're suddenly talking about expanding that from 30 or 50 disorders to 700 something disorders, we need to think carefully about all of the downstream people whom it might affect. So at the center of all of this is, of course, the baby themselves, who I think sometimes we overlook because they are a newborn when we're talking about newborn screening. But this information is something that they will carry with them for a lifetime. And so while they do have a right to health, we also need to think about what type of right they have about their ownership of their own genomic information. So that's my baby here right in the middle. Not my own baby, sorry. This is a stock image. That the baby is in the middle of all of this. And then additionally, we have to think about the way that, this actually affects the newborn screening labs, how this affects their workflows, how this affects the pediatric workforce, how this affects people like medical geneticists who are small in number already, and other specialists too, who are going to be caring for a wide range of these children. Pharma companies that are developing some of the very specialized therapies for these disorders, rare disease communities, different communities and different individuals may feel differently about early diagnosis, about wanting to label children with a diagnosis before they develop symptoms, and all of that can be very important when considering who to identify. And then people like insurers who are thinking about long-term health risks. And then, of course, parents of these infants who are going to be getting information that could be complex, confusing, in some cases life-saving for their infants very, very early on in life. And we need to think about the kind of psychosocial ramifications for them as well. So I'm first going to focus on parents and a study that we did that's currently under review. So this is unpublished data. What we did in this study was we invited mothers who were residing in 3 different locations, in Boston, in New York, and Alabama. They were from diverse racial, ethnic, and socioeconomic backgrounds to participate. And we invited them to participate in a series of interviews and focus groups. And what we were trying to understand was really, what do these parents think of this idea of genomic newborn screening? And in particular, what types of disorders do they think it would be appropriate to screen children for? So all of them had young children. And what they all agreed upon, what I haven't even shown here because it would all be green, is that they really all agreed upon the idea that we should identify childhood onset disorders that have some sort of targeted treatment. They said, certainly, how could you withhold that information from us if there is something that could help my child early in life so that they wouldn't have symptoms of a particular disease, or their symptoms might be less, then we need to be screening for that. But we also asked them a lot of really nuanced, difficult questions, which we explained in detail and provided hypothetical situations for. For example, something that a lot of geneticists and experts in this area disagree about, which is childhood non-treatable disorders like neuroregressive conditions. So would you want to know that a baby who appears healthy at birth and maybe is initially meeting their milestones might eventually develop symptoms of a neuroregressive condition that we can't actually do anything about? So in this hypothetical situation, we essentially described the symptoms of something like Rett syndrome and said, would you want to know this if there were no treatment that could be applied at that time? And what we actually found out was that most of these mothers had a positive response. So you can see 18 out of 20 of the participants in the interviews, and this is separate from the focus groups, but essentially responded positively to this question. So one quotation from a mother you can see here is, "I wouldn't like to think, why doesn't she, my child, do those things that other kids her age do instead of advancing? Why, instead of advancing, is she falling behind? So I would like to know not to expect things that will not happen, not to compare her with other children, and not to doubt myself, wondering what I'm doing wrong that makes her fall behind." This study was led by a trainee of mine who's now an attending medical geneticist, Dr. Jacqueline Omarodian. And one really interesting insight that Dr. Omarodian made after some of these interviews was that clearly our definition as physicians, as researchers, of the clinical utility of testing was not necessarily what parents were actually using. There was a different type of personal utility that they were defining, that this information could be valuable to them, to their well-being, to their understanding of their child, to the parent-child relationship, even if there wasn't a targeted therapy. And interestingly, whereas some of the responses to some of the other hypothetical situations were not quite as positive as they were to the childhood non-treatable disorders, things like adult onset treatable conditions were actually also mostly positively regarded by these parents as well. And of course, there's bias, right? They're talking to, even though we try to be unbiased in our interviews, Dr. Omarodian and I and some of our other interviewers are, of course, doctors or people who work in this area. And so, our participants may have been influenced by that. But I like to think that, we gave them the space to think for themselves and speak freely. And many of them felt that there was value in having this information, which really even diverges from our concept of just including childhood onset treatable disorders in newborn screening. Even really complex concepts like low penetrance conditions, and then we had another question about variants of uncertain significance, were well understood by our participants as we provided education on them. You can see that that's kind of reflected, the nuance of this is reflected in the various responses. Some mothers said that, for example, for a low penetrance condition, a variant that, may cause risk for a disease, but that the disease itself may never develop in their child, some of them wanted to know about the risk because it might change over time, which I thought was really interesting, that they would, want to be kind of updated, receive new information based on that genomic finding. And then others, said what we maybe might more expect, which is sort of, if it's a small chance, why should I worry about something that's unlikely to happen to my child? So in addition to that qualitative research, we've done some quantitative research as well, exploring the views of medical geneticists and other rare disease specialists. And in this particular project, we actually did a survey that was sent to over 260 individuals who all specialized in rare disease in some way. So essentially, these included medical geneticists, many of whom are program directors of medical genetics residencies. And then we also had, as authors of this project, what we called clinical champions. So we had people who were expert in rare disorders in each one of these clinical domains that you see listed here, for example, cardiovascular genetics, endocrine genetics, et cetera. And then they nominated 10 to 20 individuals in their own field whom they felt we should also be sending this survey to, who sort of cared for a lot of children who had rare disorders in that particular area. So we surveyed about 260 individuals in total, and we had created a list of over 650 genes that were all considered to be treatable, and they came from all of these different databases, the RX Genes database, which I've already mentioned, the Treatable ID database, which includes mostly treatable metabolic disorders, and then some others as well. And then we asked them basically what they thought was most suitable for genomic newborn screening. So they only had to answer in the areas that they were most familiar with. So for example, a cardiovascular geneticist didn't have to answer about cancer or anything like that. They just checked off the areas that they wanted. And we really then just basically tabulated what they agreed with or disagreed with. Because there were so many genes, we didn't collect individual responses about why they did or didn't select each one of those genes, but they were able to offer free text responses as well. And basically what you see here is that there were some genes that everybody agreed should be included at the kind of 90 to 100% concordance of individuals who answered for that gene. There were some that everybody agreed should not be included that were sort of down on the left-hand part of this x-axis, and just had maybe, 10 or 20% agreement for individuals who answered about that gene. And then there was a big gray zone right there in the middle. The most, common finding was that there were, there was disagreement basically about whether or not to include particular genes in genomic newborn screening. So one of the major takeaways from this particular study was that there were 8 genes that emerged with 90% or greater concordance across all of the participants who answered for that. So although we had, over 250 participants, they basically didn't answer for every gene, like I said, because they were able to select the particular areas that they were most familiar with. And so, for example, that's why there's a denominator here of 62 for OTC deficiency, because those were the experts in inherited metabolic disorders who answered that. But you can see that, there are many disorders here that kind of were widely agreed upon, that people felt were sort of ready for implementation in genomic newborn screening now. And what's interesting about these particular disorders is, for example, OTC deficiency is a disorder that is very well characterized, and we've tried to put it on biochemical newborn screening before. Some states have trialed that, but the particular glutamine citrulline ratio that has been used for that is not always accurate and has produced both false positive and false negative results. And so many of our experts wrote kind of in their free text comments that using genomic screening might be a more effective way to ascertain both males and females, as this is an X-linked disorder, who are at risk for that, so that we could make sure that we apply targeted therapies if needed, which can be life-saving in the neonatal period for severely affected males, and also make sure that we identify girls who are at risk so that we can make sure that they are, identified early if they manifest symptoms, or that we can avoid triggers for decompensation in those individuals. Similarly, disorders like glycogen storage disease 1A and 1B can manifest very early in childhood and can have devastating consequences, including even death from hypoglycemia, but don't necessarily have one single reliable biomarker. And so right now, the way that we perform newborn screening wouldn't necessarily be a reliable way to ascertain those individuals, because we sort of now kind of identify them based on a combination of variety of labs, of their history, and exam findings, and in this case, using genomics and a genotype first approach might actually help to identify these kids before the first episode of hypoglycemia. So many of these just sort of kind of extend the type of screening that we're already doing with kind of similar ideas, but they also are things that right now are not particularly well captured by the techniques that we're using. So what we've also begun to think about is, newborn screening labs, right? They are going to need some guidance about, how to prioritize these disorders. The Massachusetts Newborn Screening Lab, for example, has been thinking about, how can we integrate genomics? They already use it as a second tier tool, for example, for the identification of some disorders. So if a biomarker is abnormal, many state labs then proceed to using genomic testing testing as a second-tier test to better understand if someone, for example, has a pseudodeficiency allele, which may sort of give a false positive result on biochemical testing, or understanding if somebody has one of the classic variants associated with disease or may have something else. But the newborn screening labs now that are beginning to consider sort of a genotype first approach or using genomic sequencing as a first tier test are going to need some ideas about what first to implement. So there are a lot of programs that are studying this. There are over 30 programs worldwide that are exploring genomic newborn screening in some way. And some of the most well-known programs are, I'm involved with the BabySeq program in Boston, which was the first to do this and is the only randomized controlled trial of genomic newborn screening that I know of. And then some of the other really well-known programs are Guardian, which was started in New York, the Early Check program in North Carolina, and of course, Genomics England is now launching a massive kind of public-private newborn sequencing program as well. So what we found, we basically compiled all of the genes that are being screened by all of these programs, and this was led by a really brilliant medical student in Belgium named Thomas Minton, who also happens to have a PhD from the London School of Economics, so came up with a lot of really brilliant analyses for this project. Basically, what we did was we compared the content of the lists, the gene lists that are being used by 27 of these research programs that we were able to get their list. And excuse me, it's 27 programs, but it's 20 research and 7 commercial products as well that are being offered as sort of a genomic screening tool. And what we found, basically, you see here, a lot of this heat map is basically dark purple. What we're looking at here is what's called the Jacquard index, which really looks at similarities or overlaps between the lists. So the lists are all of different lengths, which affects this as well. But essentially what we found is that there is a fair amount of similarity among the commercial products, as you can see in the upper right-hand corner. So, basically all of these programs are kind of listed twice, once on the x-axis, once on the y-axis, and broken down here into the research studies on the left or in the bottom and the commercial programs on the top and in the right. And so you can see we're a little bit more yellow and green in the upper right-hand corner. The commercial products are a little bit more similar to one another. And part of the reason for that is that many of them are using just basically genes associated with the disorders on the recommended uniform screening panel, whereas many of the research programs go further in sort of exploring other treatable disorders. Everyone has sort of independently constructed their list, and even if they have similar stated criteria, they've made some very different decisions about what they're actually going to include in their programs. So I have 2 little kids, and this is one of the books that they like to read called We're Different, We're the Same. So when I looked at this data, I thought, okay, we're all really different. Like, what kind of statement can we make about that? But there are a lot of commonalities among the programs as well. So there are, basically one of the biggest predictors of whether or not a gene was included across these lists is whether or not it's associated with a disorder that's on the recommended uniform screening panel as well. We basically, have all agreed that that's kind of the status quo. Those are very well characterized disorders. They're on there for a good reason. But sort of looking beyond the RUSP, there are 34 additional genes that had 75% concordance or inclusion across the programs, which could also be considered kind of high priority targets for how we're going to expand these in the future. And so these red asterisks on the left correspond to genes that you also saw in the prior study as having very high concordance in that survey of rare disease experts. So there is consistency as well. It does seem like, for example, we all agree upon those genes, but then there were additional genes as well that were thought to be valued by these programs. So the question is why, right? The programs all sort of say, oh, well, we want many, early onset conditions. We want treatable conditions. Some of them, like BabySeq in Boston, where I am, are also saying we are looking at adult onset conditions. We are looking at things that are not treatable. And that's one of the stated aims of the program. But most people are saying we're trying to look for things that actually could be on newborn screening in population screening in the future. So there are a lot of characteristics by which we can kind of classify these genes and disorders. Is the disorder treatable? Is it early onset or late onset? Is there a non-genetic confirmatory test? For example, can we use some kind of biochemical test or use an immunologic test, flow cytometry, to confirm or to refute whether someone has this disorder? Is it thought to be of low or high penetrance? We often don't know. And is it thought to be, mild or severe disease? These are just some of the characteristics by which we can classify these disorders. So what we did in this project was we basically developed a database with the kind of 4,200 plus genes that had been included in one of the genomic newborn screening programs that we were studying. And we made a database that basically analyzed across 25 different characteristics, kind of what were these genes? Were they associated with some kind of treatment? Were they a childhood onset disease? And that data was all pulled from existing databases. And then we used that information to determine what the predictors of inclusion are for genes across these newborn screening programs, across these 27 programs. So like I said, being on the recommended uniform screening panel, that's already one of the greatest predictors. Interestingly, those don't all fit the Wilson-Youngner criteria. If you're a metabolic geneticist like me, I trained at the Children's Hospital of Philadelphia, and one of the teachings that I learned was something like maybe 3-MCC deficiency doesn't actually belong on newborn screening. The phenotype is highly variable, and most of the time those kids are healthy. But everybody kind of included the gene anyway in their newborn screening panel, so it's sort of upholding what we're already doing. Those answers from the survey that I described were also highly predictive of inclusion. And then in terms of gene and disorder characteristics, whether we knew a lot about the gene, which has been previously a previous metric that's been encoded by one of the North Carolina studies, whether there's high treatment efficacy, high penetrance, et cetera, all of these things were also predictive when we performed a regression model. We also developed a machine learning model, a boosted trees model, to basically understand, if we could predict if a gene was highly included across programs. So we trained it on 70% of our data set and then tested it on the remaining 30% of genes. And it performed quite well, actually. So you can see our boosted trees model here has an area under the curve of 0.917. And so we realized this was a pretty good model. We then applied it to all of the genes to basically rank them in importance. So which was sort of most highly predicted by the model as being included across programs would be the most highly ranked. And you can see here, these include genes from the RUSP, but for example, these are all the disorders that sort of rose to the top. What's interesting about this model is that it gives us ideas of sort of not only, things that are just included highly across programs, but because it is able to predict the characteristics of why the genes are included, it actually picked out a bunch of genes that probably should have been heavily included across programs, but maybe weren't as well kind of known to the people designing these studies. For example, several genes that were associated with inborn errors of immunity are childhood onset, highly treatable, well characterized, but only appeared in a minority of studies. So using a machine learning algorithm like this would have to, it can basically create an idea of genes to prioritize for population-wide implementation, which could then be curated manually by an expert. So in addition to genes, right, genes are going to change over time. What we know about genes is going to change. The treatments that we have for genes are going to change. So the ICONS group, the International Consortium on Newborn Sequencing, is an international group that brings together many of the leaders of those studies on newborn sequencing that are being done. And we just completed a Delphi questionnaire trying to understand how can we update the Wilson-Youngner criteria to sort of suit the nuances of genomic disorders and genomic screening. So you're getting a little sneak peek here. We're presenting this in October at the ICONS meeting. But we have come up with some ideas for sort of how to update the Wilson-Youngner criteria that address basically just much more specific ideas about genomics. Like if only variants of uncertain significance are found in a gene, no variants should be reported to the family of that infant, for example. Whereas the group was very much undecided on if there is one pathogenic or likely pathogenic identified in an infant and a second variant of uncertain significance, should we be reporting that? They may be at risk for a disease. Should we be trying to get them back to clinic to do additional studies, et cetera? So this is really only the beginning of kind of drilling down on our recommendations for population-wide implementation. So my last few minutes here, I'll just talk about how biobank data and electronic medical record data can inform the sensitivity and specificity of genomic screening. So one question that a wonderful trainee of mine, Sarah Bick, recently asked is, how sensitive is genomic testing for treatable inherited metabolic disorders? So she did something pretty simple. She actually just looked at all the records over 5 years of individuals seen at Boston Children's in the genetics and metabolism program. And she only included those who clearly had a biochemical and clinical diagnosis of a specific metabolic disorder and had also had genetic testing. And what she basically asked was, these are people who are, who've been labeled with metabolic disease by a metabolism doctor and are being followed for it and have had testing, and how many of them actually had positive genetic testing, so that if we had approached it as a genotype first screening project, how many of them would we have found? And so she found that for conditions that are on the recommended uniform screening panel, genetic testing was positive in 82% of cases. For the non-RUSP conditions, it was positive in just 76. So this means, right, we are going to miss people with this type of approach. We're always going to have to be alert to the possibility that other individuals are out there who are affected and were not ascertained through genetic testing. Of course, genetic testing for these people was limited. Some had panels, some had single gene testing, some had exome, some had genome. It really varied. And so if we were to use, for example, a whole genome sequencing in every infant, we would probably have higher sensitivity. But this was sort of using, current approaches of what we had. And it varied by disease type, but interestingly, to my surprise, patients who had fatty acid oxidation disorders were most likely to lack a genetic diagnosis in Sarah's study. We don't exactly know why that is, but that came as some surprise to us. Then another question that we asked is, how specific is genomic testing for treatable disorders? So basically, do all people who have, a variant in a particular childhood onset treatable disease gene actually go on to develop symptoms? And so in this study, this is a little bit older now, but we used the Nomad database when it was still like version 2. So it had about 100,000 sequences in it. And we had a list of disorders that could be treated with an early stem cell transplant. So at this time, SCID had been somewhat relatively added to the newborn screen. And the purpose of SCID being added was that if you did a stem cell transplant early, or for MPS I, if you did a stem cell transplant early, you could ameliorate symptoms of that disease. But there are a lot of diseases like that. So we asked, okay, so what about those? And how would you screen for those genomically, and what would you find? And what we were asking were there people in the Nomad database who are theoretically, adults who are supposed to be healthy, who don't have signs of rare disease, and would they have flagged on a genomic newborn screen because they would have pathogenic or likely pathogenic variants in these genes? Would we have identified them and, maybe incorrectly wanted to apply something like a risky, costly therapy like stem cell transplant to them? So we found, a relatively small number of them actually had, had variants that were associated with these disorders, but there were some people there. So we found, like 1 in 2,400 people in Nomad actually had some sort of risk allele, and it just begged the question, like, why? Were they really at risk? Some of them, for autosomal recessive disorders, we can't phase the variants, and so we didn't know. The variant co-occurrence tool was not developed, I think, at the point that we did that analysis in Nomad. We didn't know. Maybe these were just in cis, but that still would probably flag on genomic newborn screening, because right now most people are talking about just sequencing the infant alone. So this led us to kind of think about, are we going to more deeply phenotype people? Are we going to better understand people who seem to have variants that put them at risk for severe childhood onset or severe treatable disorders, and better understand whether or not they really manifest symptoms? So in our own hospital, we actually have a program to recontact individuals who have pathogenic and likely pathogenic variants in the ACMG secondary findings genes. So we were already recontacting individuals with variants in OTC and GLA, which is associated with Fabry disease. And then we also partnered with the Penn Biobank, who had a lot more exome data at the time of this analysis. And we actually were surprised to find that like 1 in 19,000 participants for each of the disorders actually was affected. So while that's a relatively small number, when you start to think about screening people for hundreds of disorders, we're probably going to find a lot of people who have variants and didn't know that they were at risk of disease. And so what we've begun to do is use the top genes from that survey that I presented. And so we looked at the top 54 genes, which had 75% or more concordance. And we've looked in our own biobank, the Mass General Brigham Biobank, to identify individuals who are followed at the hospital for any reason, who have given a sample to the biobank. And we have about 60,000 exomes now, and we have found 70 individuals across 54 genes, so over 1 in 1,000 people, just looking at 54 genes, who have pathogenic or likely pathogenic variants in the expected mode of inheritance for these particular genes. Meaning, if we start screening for a lot of things, we're going to find a lot of people who flag as positive. So we're now in the process of recontacting these individuals, inviting them for deeper phenotyping visits in clinic and better understanding whether they lack a diagnosis but really have symptoms, or maybe were misdiagnosed, or maybe they're asymptomatic and these disorders are not necessarily as penetrant as we expected. And if that's the case, that will, improve the way that we counsel parents if we were to find that their infant had a variant in one of these genes. So that's the plan. We're going to curate these variants. We're going to recontact people, invite them for deeper phenotyping, and use that to kind of make sense of penetrance, and then hopefully extend this to other genes in the future for other investigators in the hospital. So this is my daughter at the Museum of Science in Boston, thinking about kind of the future of genomic screening. And so my conclusions here are, newborn screening, as Dr. Buckner described, has this amazing history. We're really standing on the shoulders of giants. It's been a highly successful public health program. But now that we have genomic sequencing, we can find infants at risk for more treatable disorders. But we need to develop some consensus among all the various stakeholders in this process about which genes to screen first. And it seems like we maybe do have some core genes, at least, that we can prioritize, that experts, researchers, parents agree upon. And over time, we can begin to understand the characteristics of genomic screening by using all these big data sets that we have from biobanks. And we can, deeply phenotype individuals and understand if they develop symptoms of disease. So thank you to my many, many, many collaborators who really make all this work possible. And that's it. And that's my email address. So thank you so much for having me today.
Thank you, Nina. That was lovely. And as a reminder to people, we are going to hold questions until after all of the speakers have spoken. Our final speaker is Dr. Gonzaga-Jauregui, who is an assistant professor and principal investigator in the Laboratory for Mendelian Genomics and Precision Medicine at the International Laboratory for Human Genome Research. Dr. Gonzaga-Jauregui earned her Bachelor of Science in Genomic Sciences at the National Autonomous University of Mexico. She then went to earn her PhD in Molecular and Human Genetics from Baylor College of Medicine in the lab of Dr. Jim Lepsky. She continued her training as a postdoctoral fellow both at Baylor and at Duke College of Medicine. She's an author on over 100 peer-reviewed scientific publications and an inventor on several patents and patent applications. She's also a passionate advocate for science education and literacy through her participation in many programs, including DNA Day and Skype a Scientist. Dr. Gonzaga-Jauregui's research focuses on family-based analyses of rare and common genetic disorders to identify pathogenic variation and novel genes associated with disease to better understand the molecular architectures and pathophysiology of genetic disorders. And we're thrilled to have us here today to tell us about some of her work.
Thank you so much, Amy. Thank you for the invitation. I'm going to share the screen. Can you see the screen there?
Yes, it looks great.
Great, thank you. Yes, thank you again for the invitation to this special seminar, especially in the context of Newborn Screening Awareness Month. And I mean, the two previous speakers were great and just sort of getting an insight into the background and the personal side, right, of someone like Dr. Guthrie, who was so instrumental in the development of newborn screening, and then how this has advanced to now. And then to Nina, I mean, a lot of what she mentioned is it's great because that also touches on some of the things that we'll talk about. So today I'm going to focus on specifically the challenges and opportunities that we have for newborn screening and early diagnosis of rare diseases in Latin America. As this has been mentioned, so newborn screening is a public health strategy that can help us identify potentially serious disorders in newborns before the symptom onset or early enough so that we can have effective therapeutic interventions and reduce morbidity and mortality. Unfortunately, and despite newborn screening being a most successful public health strategy, second only to vaccination, the implementation of newborn screening around the world is very uneven. And the different conditions that are screened are also very variable between and even within countries. And we have a range from countries that have no programs even to date for newborn screening to some programs that screen for one condition and other programs that screen for up to 72 conditions. So expanded newborn screening is primarily based on biochemical screening and tandem mass spectrometry. So it was mentioned before, the very first assay that was developed for newborn screening was for PKU, for phenylketonuria, and that was the bacterial inhibition assay for that Dr. Guthrie developed in 1963. And in the case of Latin America, so even though the region encompasses 33 countries, only 16 countries have some national or regional NBS program. That means that about 7.2 million newborns are screened per year, of about 9.3 million newborns that are born each year in the region. And only two countries, Uruguay and Costa Rica in Latin America, have expanded newborn screening as a national program that screens 28 conditions. The rest of the countries have programs that screen from 1 to 6 or other variable number of conditions. And the most common condition screened across the region is congenital hypothyroidism. So here, Nina mentioned the programs in the US. So although in the US, there are recommended disorders by the ROSP, this is also variable among states, and this ranges from 28 to 73 screen disorders in different states, which also means that patients in the US can be screened in one state for a condition, or were born in one state and weren't screened there, and then they moved to another state where they could have been screened, but now they develop a disorder that could have been preventable. Similarly, in Europe, the most recent newborn screening program was developed in 2000, and also the number of conditions that are screened for are very variable, from 1 in Montenegro to 29 in Austria. And then similarly, there is variability among the different provinces and territories in Canada, with only 3 conditions screened across the country. And as you can see here, while Europe, US, and Canada started newborn screening in the 1960s, shortly after Dr. Guthrie developed the PKU assay, In Mexico, we started newborn screening until 1974. So 11 years after the first assay developed by Dr. Guthrie. And this was mainly for toxoplasmosis, congenital hypothyroidism, and PKU. And then PKU was dropped from the screening and then brought back later on. And from there, you see the different years in which programs for newborn screening have been developed in Mexico. And again, very variable from 2 disorders to 6 disorders and 28 disorders, as I mentioned, in Uruguay and in Costa Rica that have the most comprehensive national newborn screening program. To mention that in some Latin American countries, there are a few pilots and a few programs that are more restricted based on regional or hospital even, hospital system based. And of course, for people that have access to private screening, they could also pay private laboratories for expanded newborn screening. But nationwide, for the population, these are the national programs that exist. So again, there's inconsistent implementation and coverage of newborn screening and expanded newborn screening and early diagnosis programs in Latin America. This also translates in that there's very little legislation and regulation around rare diseases and access to diagnosis, treatments, and drugs. And also, there's not a consistent definition of what a rare disease is across the Latin American Caribbean region. 10 countries have no definition or legislation, and some countries have followed the definition of 1 in 2,000 individuals, that is the European definition, and others use different definitions. So in the case particularly of Mexico, this is interesting because Mexico has a very heterogeneous and complex healthcare system where the benefits and the services that people get, and as a consequence, newborns that are dependents of their parents, depends on the employment affiliation of parents. So we have 7 major Mexican public healthcare providers that tend to different fractions of the population, as I mentioned, depending on employment. And you can see here that for some of them, especially the smaller ones, such as Pemex, which is the national oil company, Sedena, which is the military healthcare service provider. This is for Navy, SEMAR, Navy affiliates. So they are already implementing expanded newborn screening in their healthcare systems. However, for the largest populations that tend to most of the people in Mexico, so we still have 6 conditions screened officially in Mexico through the Secretaría de Salud, the Ministry of Health, and then the Social Security system screens for 7 conditions. So again, very variable and uneven implementation considering the large population that we have in Mexico. As I mentioned, the first implementation was in 1974, 11 years after the first assays for newborn screening. And also, again, so only 6 conditions are mandated. So later on, there was a recommendation to include additional conditions and have expanded newborn screening, but this was never mandated. So ultimately, the different healthcare systems in Mexico decide which conditions to screen for based on their resource availability and also their technical capabilities. It's interesting to mention that despite all of these limitations, advocacy by civil organizations has been changing the landscape of expanded newborn screening. So there have been organizations that have been really advocating over the last years for inclusion of additional conditions in expanded newborn screening, starting with different states. So the states of Guanajuato and Quintana Roo have already approved in their state legislation to do expanded newborn screening, and sort of slowly trying to get these to the federal level so that it's nationwide. But one of the successes in that are more recent in terms of raising awareness and also making this a more visible need nationwide is the inclusion of the results of newborn screening in the national child wellness card, which is a card that every child in Mexico gets, where vaccinations are recorded, and they track wellness visits and any health-related items throughout their first few years of life. So in this case, the expanded metabolic screening, so these referring to the 6 conditions that are officially mandated by the government, should be included here. When was it done and what was the result, if it was positive, negative, whatever that was. The hearing screening, ophthalmologic screening, cardiac, and hip dysplasia screening. So all of these screenings should be included now officially here, and that has to be tracked. And this is important because one of the major issues of newborn screening programs in Mexico and Latin America is that many times they take the sample for the baby. The parents knew that the baby's heel was pricked, but they never got a result. They never knew what was, if they had a condition or not, like they were never notified. Another thing that we are also trying to do in collaboration with civil organizations is, again, informing the population that this is a right they have, because even for the 6 conditions that are mandated, this is something that every baby in Mexico should have. So we have also been part of International Newborn Screening Day and producing informational materials to raise awareness about the importance of newborn screening and knowing the result of newborn screening. And so, as I was mentioning, this awareness and also this tracking of the results is really important, because newborn screening is it's not just a test, but it's a whole process. That includes several steps, including pre-test information. So informing the parents before they even have a baby that this is a right, that this is something that will be beneficial for their health of their baby, and what are also the risks and what are the limitations of the test. Of course, the part that we focus probably the most is the screening and ensuring that this screening is done properly, that it's done in the time frame, that it's the best to detect abnormalities in the metabolites or whatever the testing method may be. And also considering other issues of diet, transfusions, prematurity, or anything else that might affect the screening process. Most importantly, as I mentioned, the follow-up to this screening, because again, many families don't know the result of the newborn screening. They know, many of them in Mexico, we've done surveys on these, they know that the heel prick test exists, but they actually don't know what it entails and what are the results of their babies. So this is really important. And also, and of course, in terms of patients or newborns, they have either inconclusive or positive results from newborn screening, do the appropriate follow-up and the confirmatory testing that is needed to actually have a diagnosis for the specific condition. And then also, of course, the treatment and management, that it's appropriate for the patient that has the corresponding condition, ensuring that they have access to treatment, because again, if we are detecting these conditions and not treating or managing these patients appropriately, then we are losing that opportunity that newborn screening is providing us. And also, finally, monitoring of the newborn screening programs, which is a constant and ongoing process, evaluating all the stages of this program and tracking the effectiveness, the coverage, and also the outcomes of the patients that are screened. Unfortunately, throughout all this process in the newborn screening journey, we have issues in Latin America that curtail our ability to really apply this strategy effectively to prevent morbidity and mortality for children in Latin America. And so one common thing that we see for patients with rare conditions is this patient journey that it's very convoluted, as you see here, and that is often referred as the diagnostic odyssey. And one of the things that we have seen for this diagnostic odyssey is that prior to 2010, the average of time that it took to obtain an accurate diagnosis for patients and families with conditions that are most often genetic was around 10 years. And in the recent years, what we see is that in countries or in systems that have implemented genomic diagnostics for the study and the diagnosis of patients with suspected genetic disorders, this has really reduced the time by half or even less than that to about 3 years for patients that present symptoms, and then they can get rapidly tested. So we know that genomics has really revolutionized the way we study, we research, and we diagnose rare diseases. So prior to 2010, so here in orange, you can see the different numbers of genes associated with different genetic conditions that were published per year. And then in 2010, when we started doing genomic sequencing, and then the development of exome sequencing, which really skyrocketed the number of patients that could be tested for genomic variation causing disease, these really produced about 250 genes per year on average that have been published since then, associated with some of them, many of them reported genetic disorders, but also many of them new genetic conditions that haven't even been reported in the literature, and many of them that occur at very low frequencies in this ultra rare spectrum. But given this availability of genomic sequencing and the reduced cost of genomic sequencing in the last few years, this has really empowered the community to study these conditions and, most importantly, diagnose these patients. So about 7,000 conditions have been reported as rare diseases, and we know that 70% of them start in childhood. And we also know that more than 6,000, so last time I checked, about 6,800 rare diseases have a known genetic molecular cause. And this is very important because this means that we can actually diagnose them. We can perform genetic testing, genomic sequencing, and we can find the cause of these diseases. And ideally, for some of them, we could find this cause early on and before symptom onset. And so, again, this is important to have early interventions and avoid adverse outcomes, such as maybe surgeries or treatments that are not recommended for some patients, depending on the specific genetic condition that they have. Also, access to effective and personalized therapies that are designed to address the molecular defect that that particular patient have. Also, it enables future planning based on the disease prognosis and the information that we have on natural history and how that will develop, and provides accurate answers for the parents and the families, enables genetic counseling for family planning, if the family wants to have additional children, or even for family counseling if there are conditions that also might affect other family members and they are not aware of. And ultimately, this allows the development and implementation of public health policies that are based on disease prevalence. But this is very important to do based also on the different populations, because disease prevalence will be different across different populations. And ultimately, the idea is to enable the implementation of precision medicine, both at the individual, but also at the population level. So genomics has really brought a paradigm shift in how we diagnose and also manage rare diseases. So in the classical paradigm, where we start with a clinical diagnosis that focuses on the clinical signs and symptoms that are manifested by the patients, this can be subjective because it depends on the knowledge and experience of the clinician. And of course, I don't think there are many clinicians that know by heart the 7,000 or so conditions that are out there. So these sometimes delays the diagnosis because the clinician might not be familiar with this specific condition. And then the patients are sent to have a lot of testing, many of which will be inconclusive. Obviously, that also takes a long time. And eventually delaying their obtaining a diagnosis. And ultimately, this is a reactive approach because it is evaluating and treating the symptoms and problems that are already present in the patient. With genomics, now we have this opportunity to have a molecular diagnosis that relies on the analysis of the genetic and genomic information of the patient. It can be more objective because it's independent of the clinical knowledge of the physician. Of course, there's a need to have this conversation with the physician to follow up on any findings that are there. But we rely more on the information that's available more broadly in the databases rather than on a specific person having heard or seen that specific condition in med school. And one test can potentially identify the diagnosis rapidly and accurately. And again, most importantly, it is preventive because we can start identifying diseases and predicting the symptoms before they even manifest. And this is very important and very evident for newborn screening because this is exactly the principle of screening newborns early on. Unfortunately, in Latin America and other low and middle income countries setting, this availability of doing genomic testing and other molecular diagnostic testing is impaired by different factors, such as the high cost and the low availability of these testing technologies in the countries. This also results in low testing demands because, again, there's low availability, but also physicians know that patients cannot pay this, and then they don't even tell them that they could get an accurate molecular diagnosis through some molecular testing. There's also a lack of awareness from primary care physicians and, of course, from patients. And then there are a few specialized laboratories that have limited training and limited technical support for running these technologies and these instruments. Again, there's poor policy maker support due to also lack of awareness about the effectiveness of these technologies and the cost benefit, right? Many of these technologies are seen as very expensive, and then many policymakers think that this is something that can be done elsewhere in the US, in Europe, but not in Latin American countries. And therefore, there is no interest in supporting legislation and regulation that can guarantee access to genomic testing for patients with suspected genetic disorders. And again, the part of the limited testing is also, as I mentioned, the cost, because this is not covered by the healthcare systems, which are mostly public, and very few private insurance companies in Latin America are thinking about this testing as an investment for the health of their insurers. Here, and as Nina mentioned before, so we have started already seeing that beyond the diagnosis of patients that already have genetic diseases, now we're seeing a lot of genomic newborn screening programs and pilots that are starting to evaluate the effectiveness of the application of genomic sequencing in the newborn period, or in the very early life, like the Baby-Seq project. So here is a map that was published last year of where are these programs, and Nina also mentioned several of them. So in the US, we have different programs, Guardian, Baby-Seq, Begin NGS, but also Europe, England, of course, with the Generation Study, Screen for Rare, Baby Screen, and other programs. But again, as you can see from this map, most of these programs are happening in high-income countries where these technologies are already being adopted. They have already a history of showing effectiveness for the diagnosis of rare diseases, and now the exploration of these application to a population newborn screening. But then what happens with countries in Latin America, in Africa, in Southeast Asia, right? So we continue to have this problem of representation, of access to technology, and of equity. And this is important because genomic newborn screening really has the potential to enable precision medicine throughout the lifetime, from very early on in the newborn period, but also potentially that information could be used and leveraged throughout the lifetime of a person, and even for conditions later on. And I'm not saying to return adult onset results for a newborn, but potentially, and this is one of the issues that it's also being explored, right? Whether to save that data and utilize it at different stages in life as these newborns grow and develop, and explore how else can this be informative. I think we are at a really interesting point in genomics, in precision medicine, and of course in the application of newborn screening, where we have that opportunity to really realize that promise of precision medicine. Again, we have this issue of representation because we know that we don't have a lot of diversity in genomics. So more than 90% of the available genomic data is from European ancestry individuals, and only 0.34% is from Hispanic or Latin American individuals. And this has implications that are real for how people benefit from these projects. Because we have increased disparity in the access and benefits of genomic discoveries for these underrepresented and underserved communities. We know there's a lack of transferability of association markers for complex traits among populations. And also we have a lack of specificity in genomic interpretation of rare and medically actionable variants for individuals from these underrepresented populations. And this is including for interpretation of variants identified from genomic newborn screening programs. So we need to increase this diversity in genomics studies, but also we need to make it in an ethical, respectful, and inclusive way that also benefits the populations that this data is coming from, not just for the sake of improving diversity, but also having a commitment to making these results available to those populations so there's a benefit to these populations of origin. So this is in line with the recent work from the World Health Organization on accelerating access to genomics for global health. So after the creation of the WHO Science Council, genomics was the first topic that the Science Council decided to focus on because of its amazing impact in health and also its potential to reduce health disparities around the world. And one of the recommendations from WHO in this report that was published a couple of years ago is that genomics programs should be based on national and regional needs and are influenced by population structure, disease prevalence, economic status, and competing priorities. So as part of this focus on genomics and how this can be used for reducing health disparities, so the WHO has started to have meetings in different regions in the world to precisely assess what are some of the needs and what are some of the challenges faced around the world. And so back in May, we had this meeting in Brazil to talk about human genomics in health and enhancing the impact of human genomics in the Americas region, including also Canada and the US, but also discussing some of these very important challenges that we face in Latin America for accessing technologies and for implementing genomics for precision health and for diagnosis of patients with suspected genetic conditions. So the question that I get asked often is like, are we ready to do genomic newborn screening in Latin America? So we had a paper published earlier in June this year, exactly a perspective on well, where we are and what we need and what we hope for the region. And some of the reviewers came back with comments about like, well, so you're saying there are all these deficiencies in newborn screening in Latin America, and not even the implementation of expanded newborn screening, metabolic newborn screening based on mass spec in the region. And now you want to do that jump into genomic newborn screening. That's kind of a big jump, right? My answer to that is that, I mean, it feels like we are chasing that train, right? Like this train of expanded newborn screening and trying to catch and get on that train. But now we see this other train of genomic newborn screening coming, and we are at a really important point where we can make that decision to invest in the new technologies that will give us much more return for that investment, which is big for the countries in Latin America, but can also have a huge impact in the population. And one of the things that it's important to mention is that we have local expertise and data is becoming available. So some of you might have seen last October, there was this beautiful cover in Nature that included this particular edition, included 2 studies on large data sets from Mexican individuals, one with available array data, one with exome data available. But we're starting to have this data available to do exactly what Nina mentioned at the end of her talk, right? Leveraging these large-scale biobanks, these large-scale projects to assess what are the frequencies, what are the conditions that we see in the population. So we have that data now. And even some of these data from Mexican projects are being used in some of the pilot programs that are being run in the US because of the importance of Latin American population in the US being the most rapidly growing minority in the country. So the only difference that we have between what some of the newborn screening pilot programs are doing in the US and what we can do in Mexico and hopefully in other countries in Latin America is the production of the genomic data. It is very costly for us to produce the genomic data, and unfortunately, we don't get much traction from these large pharmaceutical companies and other biotechnology companies to form these large consortia that support these type of large-scale pilot programs like Begin NGS or Guardian or BabySeq or others. So we really need that support to show that this can be done in our countries, and this is really impactful for our population. So ultimately, I do believe that human genomics has the potential to reduce the equity gap across countries, but it can also broaden it if we don't work as a community to really reduce that gap. And this equity starts from the newborn period with now what we see with genomic newborn screening, assessing these programs, how would they work in different economies, in different healthcare systems, and guaranteeing that every patient and every child that can benefit from early screening in the newborn period can get that equal access and life opportunity. So with that, I'll just thank all the people in my lab, collaborators, and I'll be happy to take questions.
Great, thanks, Claudia, and thanks to all of our speakers. Jane Buckner had to hop off, so she sends her apologies that she needed to run to another meeting. But now I'd like to open up the Q&A for people who have questions for either of our other two speakers. And I see that Bill Hagopian, you have your hand up.
Thank you very much, Amy. This was just a fantastic presentation. You guys really need to be congratulated on the complementarity of what you've said and the breadth of it. It's amazing. But I wanted to spur you on to increase the breadth even more. So you're talking, and this is a question especially for Nina, but also maybe for Claudia. You're talking about monogenic inborn errors penetrant in infancy or later in life. But some of us also consider newborn genetic screening for more common diseases, which have a prevalence of 1 in 300 or 1 in 100 rather than 1 in 3,000 or 1 in 10,000. And the one I happen to work on is type 1 diabetes, which is 1 in 300, but also things like celiac disease, hypercholesterolemia. You know, there's a whole list of common diseases that have polygenic plus environmental etiologies. So that involves some different considerations. I mean, polygenic risk scores, usually SNPs are sufficient and you don't have to sequence. So a SNP array that has 500,000 features with an imputation against TopMed or another whole genome database that has a lot of ethnic variants in it can get, 9 million SNPs. And I believe, and then you can take a computer pipeline and generate a polygenic risk score. And I believe even maybe cystic fibrosis can be dealt with in that way because there's a lot of expertise and experience with that one. But it costs less than $40 a subject, and it might be able to slingshot us forward until whole genome sequencing is widely available. And of course, when we have whole genome sequencing, we'll get both the monogenic diseases and be able to do polygenic. But until then, SNP arrays sound like a really cost-effective way to go about it. And for a common disease, if you do genetic screening and then follow up later in life with a biochemical test, you may need to actually screen 10 or 20% of kids, which is a huge number. And it's costly to collect blood in real time to find the metabolite that's increasing or the autoantibody that's appearing. And on so many kids, that's super expensive. And that's why the genetic pre-screening makes sense rather than just biochemically test across the whole population at the age at which you might expect the biochemical test to turn positive. And that brings me to the Wilson-Junger criteria, which didn't include cost benefit or cost effectiveness criteria. And so I'd like for you to comment on the role of genetics in that. I might add that when you lose a little bit by pre-screening with a polygenic risk score and then doing a biochemical test, the product of those two sensitivities might be only 70 or 80%. But Nina, actually, you talked about other monogenic diseases that had 70 to 80% sensitivity. So we're kind of in the same boat. Of course, we don't want to let the perfect be the enemy of the good. But for these common diseases, even with a 70 or 80% pickup, 1 in 300 disease, there's a huge number of individuals that'll benefit. So I wanted to know the intractable problem is really the sample follow-up, and that's not something newborn screening does. They do screening at birth. What are your thoughts about this common disease approach?
So I think there's a lot of amazing information in that question. And so I know more about monogenic disease than I do about common complex disorders. But I do know that the eMERGE study had been looking at pediatric polygenic risk scores, including for type 1 diabetes. You may be very familiar with that work. But interestingly, just anecdotally, when I approached our own pediatric and adolescent practice at Mass General Hospital to ask them if they would be interested in enrolling patients for eMERGE, they said, what are we going to do with this? And I thought that was interesting. So one advantage that we have in the monogenic world is like, I can say, we're going to put you on a low protein diet, or we're going to put you on this particular medication, this enzyme replacement therapy, something highly targeted to the disorder. And so in conversation with them, one theoretical scenario we came up with was something like, okay, let's say, a child who was potty trained starts having, nocturnal bedwetting, and you don't know what's going on, or they aren't gaining a lot of weight, or they lose a little bit of weight or something. And then your pretest probability is a little bit higher. Maybe you wouldn't have checked a glucose, you wouldn't have gotten a UA at that visit, but you're going to because you know that they're at risk. Almost using it in the same way as you would family history, like if they had a sibling who had type 1 diabetes. But I think the advantage right now of some of the monogenic disorders versus obesity risk or type 1 diabetes risk is it's very easy to tell somebody, if you were at risk for this, here's what I would do about it. Or you are at risk for it, and I can figure out right now if you have it or not. Whereas with, being at risk for type 1 diabetes, people may be able to change behavior, change nutrition, things like that, but there's not something that they can do right in the moment to necessarily treat that risk. So I definitely agree with you. We're going to find more people by definition, right? If you pick the top, 3% of your PRS, like you have a hard-coded number of people you're going to find every time. But I don't always know that it is necessarily as actionable, but maybe if we targeted those people for dietary or, behavioral interventions, that would be a good use of those resources or something. But
Well,
you know more about it than I do, I think.
ketoacidosis goes down tenfold with education of the highest risk kids and those that are autoantibody positive. But also now there's an FDA-approved therapy, an immunotherapy, monoclonal antibody for delaying the onset of hyperglycemia. So yeah, we have stuff, finally.
Claudia, did you have some comments about that? Oops, sorry, you're muted, Claudia.
Right. Yes, I just want to mention that, again, so while some studies showing that a clinical implementation of PRS is starting to be useful for some conditions, we also know that these PRSs are not transferable across populations, and there are studies showing that the accuracy and the prediction accuracy for PRS, it's very variable depending on the ancestry of the individual. So again, this is another area where, yes, they could be very useful, but it depends a lot on what the person background is, because it won't perform similarly with a European ancestry individual versus an African-American individual. And that goes back to this lack of studies done in other populations that are really a need that we have.
This is absolutely important. In our data from the US, African Americans have a different sort of approach. The scores are still useful, but they're not quite as useful. And Asians even less, but we've done studies in Africa and in Asia that will help us make those tests better. The good news is Hispanic Americans are usually Caucasian, and the PRS scores for type 1 diabetes perform really, really well in Hispanic Americans as well as Caucasian Americans. Now, if you're in Brazil where you have people of all colors and all backgrounds, that might not be the case, but very good point. Thank you so much.
We have a question in the chat from Carolina Lapostal for Claudia. In your opinion, what would be the main barrier to increasing neonatal screening in Latin America?
I think for a lot of these public health policies and strategies, the involvement of government is so essential. And I think that's one of the major issues in Latin America. So again, in Mexico, for example, so 6 conditions are mandated. There was another legislation that was set, recommended expanded newborn screening. But again, there's no mandate, and much more important, there's no budget for the healthcare systems, the public healthcare systems to actually do that. So even if they get the recommendation, many of these public healthcare systems are quite constrained. And also they might not even have the technology, right? In this case, mass spectrometry to do these testing. So it is very important that governments are aware, and they also provide the budget that is necessary for the implementation of the programs.
And actually, if I could, so that was kind of related to a question that I was wondering about. So with respect to, the potential for countries that are doing very limited newborn screening right now, or have even not started newborn screening by mass spectrometry, is there the possibility, do you think, that these countries might leapfrog and just forego mass spectrometry altogether? We talked about not letting the good die in the service of the perfect. And I appreciate the fact that this is clinical work, not yeast genetics. Might there be some benefit for, going straight to sequencing based, and in some countries, where things like Oxford Nanopore technology might make them cost effective. And so that would be my question for Claudia. And then my question for Nina is, can you speak to how some of the comparative studies of conventional versus sequencing-based newborn screening might inform those decisions?
Yeah, I definitely think so. I mean, this wouldn't be the first example, right, of leapfrogging technology and sort of getting, even if you haven't implemented the previous one, sort of going to the next level. And I think we are exactly at that position where considering the cost benefit and the application of genomics to not just newborn screening, diagnosis of rare diseases, population screening, and precision medicine more generally, I think there's a lot of potential in that. Yes, if you make that investment in that technology, that will pay off in all sorts of different ways. So I definitely think we're there where it's important for governments to do this evaluation of that cost benefit. And we can do, and I do hope that we do that in Latin America, where we cannot keep chasing the mass spec train, right? We need to get into the next train that's coming.
Thank you. Nina, do you have any thoughts about the comparison?
Yeah, there have been some studies. There was one using the BabySeq data where they compared the people who had positive genomic screening results to those who also, underwent state-run newborn screening programs. And the traditional newborn screening is more sensitive. We are gonna miss people from a genome first approach, but it's things like hearing loss that we often miss them on, which may not necessarily have genetic underpinnings in every case. So obviously, there are cases in which kind of some newborn screening methods extend beyond what we're going to capture from sequencing alone, and from some of the work I presented from Sarah Bick, we know that's true even with regard to inherited metabolic disorders. I think as genetic testing gets better, once we're if we're using an exome, we're using a genome for everybody, we're going to capture more. And then those are mainly with regard to things that are already included on the recommended uniform screening panel. But genomic screening can obviously go so much farther than kind of traditional biochemical techniques can. So yeah, I think if I were starting a program from scratch in an ideal world, if I had to choose, there are always going to be complementary technologies. It would be great to be able to use both. But I do think you could probably ascertain a greater number of true positive cases just using a genotype first approach.
Thank you. Does anyone else have any questions, either in the chat or Claudia? Claudia?
Yes, I actually, well, thank you so much for both of you. It was a great presentation and in different aspects, right? I learned a lot. I have a few questions, or maybe comments, I guess. I was very surprised with one of the slides that you showed, Nina, where there is the high evidence for a gene causative is like only 30% of the criteria for inclusion. But obviously this showed the importance of the the selection that is already out there, right? Because that's the most important one. So the decision, it's like there is a lot of responsibility for the decisions, right? Because of this group. I don't know exactly who is this group that decides if it's European or American or but that gives me the thinking that Claudia comment about the fact that prevalence is important, and that's one of the whole criteria. But you mentioned that prevalence is not important. You guys are kind of thinking a different way. The prevalence is important. So I found interesting that the two different approaches. And also I was curious about like how the diseases are specific for certain countries. If for instance, there's something in Mexico, Claudia, that you guys are thinking about the newborn screening will be more important, or in other parts of the country that the prevalence is going to be really a key factor in choosing or test.
I don't know, Nina, if you want to start.
Yes, I think, this question about the evidence base is interesting. Some of the newborn screening programs actually did include even genes that had like refuted or limited gene disease validity on ClinGen. So like everybody is thinking about this very differently, or maybe some of our manual curation isn't as closely done as we might hope it is. So yeah, I don't know. I think everybody has a little bit different standards. Like some people, when we were kind of talking to the icons group about this, said, well, maybe you don't have a lot of evidence or something, but you could identify people at risk. And then if there's a non-genetic confirmatory test that you could apply before you give anybody treatment, then it's okay, because you found who's at risk, and then you can do a diagnostic test. So everybody has a little bit different views on it, which is why I think maybe that wasn't as, high a criteria. But that particular metric, I think, came from the Nexus study, and it might be a little bit outdated too. So somebody suggested to us recently that we could be using kind of other tools, like the number of times that the gene is mentioned in PubMed in association with the disease, or something a little bit more computational, that maybe would be a better metric of something like evidence-based. So it could be a combination of things.
Yeah, I think, we know that, first of all, right, we don't have a good characterization of variation in Latin American populations, but we also know that even within different countries, within different populations, we have a lot of founder populations. So in Mexico, we've already identified several founder mutations that are specific to certain regions, many of them happening in the indigenous component of the genome, and that's so, or mostly indigenous populations because of geographical isolation or whatnot. So, and this is why I say this is important to know the prevalence, because at some point, ideally, right, in an ideal world, we should be able to, or we would want to sequence everyone and have whole genomes for everyone. But it is also true, again, that in Latin America, we have limited resources. So maybe doing studies that assess what are the major variants that could then help design other arrays or other methods that can be used more widely for population screening could be done, but then that's why it's important to know what variants are more prevalent or which conditions, because we need to do that very tailored to the specific population. Same thing currently in several, well, not several, a few Latin American countries, glucose 6-phosphate dehydrogenase deficiency, it's a major condition that's screened for because there is a high prevalence, which is not that relevant, for example, in European ancestry populations. Cystic fibrosis is screened in Latin America, but it's not as prevalent as in European populations. So I think we do need to be aware of those differences among populations to find ways to make these strategies most effective for those specific populations. And again, considering economical constraints for low and middle income countries, having some awareness of which diseases are important to screen for, which conditions could be actionable, and we could have some early intervention and have those being screened for. I think that's very relevant.
Okay, thank you.
And of course, you know, Claudia, as you pointed out, you know, genomic sequence can have impacts for diseases that show up across the lifespan. And so, you know, we're focusing on newborn screening, but, you know, if the cost of screening for PKU, galactosemia, and 3 other things is equivalent to a whole genome sequence, then, you know, the cost benefit analysis might look very different.
Right.
Right. Well, I'd love to thank you both so much. This has just been a wonderful experience, and we've learned so much. And I really appreciate the fact that that we're going to be able to share these presentations online, so the impact will continue to grow exponentially. So thank you both very much for taking the time to meet with us today and give us all this great information and your perspectives.
Thank you. Thank you so much for the invitation.
Thank you.
All right.
Thank you. Bye.
Bye.